Hello, everyone, and welcome to the SRNA Ask the Expert podcast series, Research Edition. Today's podcast is entitled Genetic Study of AFM. My name is Rebecca Whitney, and I will be moderating this podcast. SRNA is a nonprofit focused on support, education, and research of rare neuroimmune disorders. You can learn more about us on our website at wearesrna.org. That's wearesrna.org. This podcast is being recorded and will be made available on the SRNA website and for download via iTunes. During the call, if you have any additional questions, you can send a message through the chat option available with GoToWebinar. For today's podcast, we are pleased to be joined by Dr. Priya Duggal. Priya Duggal is an associate professor of epidemiology and international health at the Johns Hopkins Bloomberg School of Public Health and the director of the Genetic Epidemiology Program. She earned her bachelor's from Wesleyan University and her master's in public health and PhD from the Johns Hopkins Bloomberg School of Public Health, and completed her postdoctoral fellowship at the National Human Genome Research Institute at the National Institutes of Health. She is the director of the Maryland Genetics, Epidemiology, and Medicine Training Program, director of the Genetic Epidemiology Program at Johns Hopkins, and director of Genetic and Genomic Analyses for the Environmental Influences on Child Health Outcomes, or ECHO, an NIH-funded study of 50,000 children and their parents. Her primary research interest is on the host genetic susceptibility to infectious disease. She works with adult and pediatric populations within the United States and internationally to identify host genes associated with parasitic and viral infections, as well as diarrheal disease and malnutrition. Her work has led to the identification of loci associated with the infectious diseases Leishmania, hepatitis C, HIV, Cryptosporidia, Shigella, and Entamoeba histolytica. More recently, her work is on the host genetics of acute flaccid myelitis and poliomyelitis. She has more than 100 peer-reviewed publications and has trained 11 current or former doctoral and postdoctoral scholars. Welcome and thank you so much, Dr. Duggal, for joining us today.
Thank you so much for having me.
We sincerely appreciate you being here. We'll jump right into our questions and kind of start with how you first learned of acute flaccid myelitis and what about it piqued your interest as a researcher.
Absolutely. So it was the fall of 2014. It was probably pretty early in September, and we had started to hear about some of these cases in the media of children who had presented sometimes with a mild viral infection, and then they were paralyzed. And as anyone who was thinking about that time in 2014, those are relatively rare. They got quite a bit of press. And my colleague, Erin Millstone, who is a pediatric infectious disease specialist, and myself were talking about it along with another colleague of ours, Dr. David Thomas, who's the chief of infectious disease at Hopkins. And we started to ask a couple of questions. Really, what could it be? What we were seeing both in the media, but also it coincided at the same time with some of our national conferences on infectious diseases. And we were asking this question of what could it be that would only affect a very small subset of individuals. The idea about it being that if it was just viral, we would have seen, lots of children that were affected with this and having paralysis or perhaps meningitis or different types of symptoms, but we weren't seeing that. We were just seeing very rare cases. And so we thought to ourselves that perhaps there might be a genetic underpinning to this disease. And we sort of set out to try to figure out if we could assist in trying to understand what was going on.
That's very interesting. So with seeing those cases in 2014, how long after that did your study formally begin?
So we actually started it pretty much immediately. We had a couple of meetings in the fall in September or so, and I think we launched our study. In order to do a research study, you have to get approval. So we got approvals from our university to get specific informed consent, to develop a medical questionnaire, and then we initiated it right away. What we did is we contacted a whole bunch of our friends and colleagues across the United States that were hospital epidemiologists, that were infectious disease specialists, and then eventually who were neurologists. and we just reached out and said, look, we're going to start this study. We don't know what this is. You don't know what this is. If you have patients that are seen in your hospital, let them know that we're starting this study and we'd love to talk to them.
Okay, that's wonderful that you were able to begin it so quickly. And what is the time frame for the study? How long do you anticipate it to go out, or when do you anticipate it to be concluded?
Yeah, so the goals of our study really are to understand the host genetics of AFM. There's sort of two big questions that we've been trying to answer in general in the field of acute flaccid myelitis. The first question is, what's causing acute flaccid myelitis? So who's getting infected, but also what's causing that infection? And many of us believe that an enterovirus, which is part of the same family that polio is a part of, is likely the culprit. But as many also know, lots of people are affected with enteroviruses. Most of the population can be infected with an enterovirus, and only a small subset of individuals actually develop paralysis. So some would estimate maybe 1 in 100,000, perhaps 1 in a million, which was also very similar to what we saw historically with poliomyelitis. So our study timeframe is really dependent on getting enough cases to understand and answer that second question. We would hope that at the end of this, we have some answers about the mechanism of disease so that we can figure out what genes might be playing a really critical role to cause such a severe outcome.
Okay, that is helpful. Thank you. And can you share anything about what you've learned from the study thus far, and maybe how many participants or families have contributed to this study? And I know you need more in order to get some answers, but do you have a definitive number of
Yeah.
what you would
Yeah.
need?
So we started in 2014, and it's a slow process because of the types of information that we need. Specifically, quite a bit of information on each child's medical records and getting some of the imaging that they had. So the first question we asked was about, could we understand what similarities there were among AFM cases in their clinical presentation? And so we worked with some wonderful neurologists, Dr. Carlos Pardo, Dr. Matthew Elric, and Dr. Olwen Murphy, who worked with us to sort of review all of the cases that we initially had seen in the first 2 years or so. And we started with about 40 cases or so that were reviewed. So each of them were blinded in their review. And they made judgments on whether or not they thought this was a classic case of AFM, which is a relatively new disease, right? How we're defining it. And so they looked for characteristics among the patients. Some of the things that we found, and has been supported as well by the Centers for Disease Control and Prevention and other individuals, is that most children had some sort of viral symptoms prior to the onset of prior to the onset of paralysis. And we have noted that. It's not true of every case that we think is AFM, but we do believe that the majority of them had that, and it was reported by their parents. So what we developed initially is sort of a research AFM criteria, something that really helps us in AFM and this is true of really any disease, is defining a set or a group of individuals that are all pretty similar in their outcome. So they're homogeneous, right? There isn't a lot of differences among them because it's likely that the same mechanism is causing disease for all of them if they all present the same way, or at least that's what we hope. So we try to define a sort of tight research AFM criteria. And we aren't excluding the other cases. We aren't arguing that if a child didn't meet exactly those criteria, they aren't AFM. But we did look through all those files to say, hey, could it be something else? Could it be stroke? Could it be transverse myelitis? Could it be another definition? That's the first step that we've done, and we've learned quite a bit about defining AFM. And now we've collected more than 100 families have been enrolled in our study, and we have started sequencing about 45 of those families, and we're reviewing that sequence information to look for similarities among the cases. So if I look at the DNA sequence of a case of AFM against another set of cases of AFM, do I find things that they share in their genetic sequence that's not shared with their siblings? Because we know that no siblings yet have been reported with AFM. And then also, really importantly, we don't really understand if this is genetic, how it's being transmitted. So there are different ways that genes can be transmitted. One is that you inherit one copy from one of your parents and it acts in a dominant form, meaning that all you need is one copy to have disease. Another way is that you can have recessive, meaning you have to have two copies from each of your parents, and then you get disease. Recessive diseases tend to be a little bit more rare and a little bit more in line with what we would expect with AFM. But there's also other options. You could have what we call de novo mutations, and that means that neither parent has the mutation, but their child has it. It just sort of spontaneously developed as the child was being formed. And what we're looking for right now is, are there any de novo mutations that are shared among all the cases?
Thank you so much. That's incredible information and really interesting to know how deep we're looking into um, how this comes about for a child. You kind of answered a little bit one of the questions about eligibility for the study. So it does not require that someone have a confirmed case by CDC's
No.
definitions. It can be a clinical diagnosis,
That's
correct?
right. That's exactly right. So a lot of cases, especially in 2018, that enrolled in our study had not yet even heard from CDC if they were a definitive case or not by the time they had enrolled. I think, early on in 2014, when we started to see the first sets of national cases, there was a lot of concern about who we were including and what we were including, and it was slightly narrower in our perspective of what it possibly could be. And then it extended much larger as more cases were seen across the United States, where it got pretty loose in its definition. And so at this point, what we care about is if a clinician has defined you as AFM, we are happy to have you enrolled in our study. We will still do blinded review of all of our cases to see where they fit into the spectrum of AFM. And that's something that's really interesting that our colleagues in neurology have noted, is that there's a really large spectrum of AFM. There are children that are on respirators, and there are children that have specific limb loss, and then there are individuals that have really recovered. And we've also started to see some adult cases. They're rare, but we've seen some of them. So it's really hard to say that we would only stick to one criteria of AFM. What we want to do is sort of enroll as many participants and try to really understand the disease and where a child or an adult may fit on that spectrum.
Okay, thank you. That's great that you do have some flexibility within there to have additional eligible, so that we're making sure that we get to the bottom of it. So if what is the time frame for since diagnosis that a family can participate in the study? I know that there are several families who were likely previously diagnosed as transverse myelitis in years past, who are now thought to likely be AFM. So is there a particular time frame from when they were diagnosed to now when they can participate? So if someone was diagnosed 10 years ago and they essentially um, appear to likely be an AFM diagnosis from what we now know of AFM, would they be able to participate in the study?
Yeah, that's a really great question. So, because we're doing a genetic study, and the amazing things about your genes is they don't really change much, right? So, the genes that you have sort of stay as they are. We don't have to worry about what we call temporality, which is the timing of things. That's not really true when we think about what we're looking at with the virus per se, but when we're looking at the host, it doesn't matter as much to us. We just want to be able to capture your DNA at any point in time. So we're okay with using older cases. We definitely have participants from 2014 that have reached out, in 2019, and that's wonderful. And we're happy to enroll those families in our study. In terms of older cases than that, I think that the way that we've sort of approached it is we're happy to talk to you. You definitely should reach out and participate because none of us know enough about what's happened with AFM to know if some of those cases really are AFM that have been misdiagnosed. The beauty of our research group is that we work within Hopkins, but we also have a lot of experts that we work with across the United States who are really dedicated to AFM. And so we're happy to review those cases and sort of make a decision on whether or not they could be enrolled in our study, regardless of the fact that it may have occurred many years before. So in a nutshell, we invite everyone into our study that has some diagnosis of AFM. And with enrolling in our study, it does mean that we would need to see those medical records and hopefully some of that imaging data.
Okay, that's great. And then just another clarifying question too. So a participant does not have to have confirmation of having an enterovirus as or another infectious etiology as suspect in AFM in order to participate. Is that
They do
correct?
not. That's correct. And that's really because most cases of AFM don't have that, especially in the United States. And we recognize that timing is really important here about that etiology. So no, we do not require it. We do try to talk to families to see if any of the children were sick prior to the diagnosis. It is, as we all know, most of these cases occurred during cold and flu season. So we anticipate that people were sick in the household. But no, it's not a requirement that you have a confirmed enterovirus.
Okay, that's great. I know that that is an incredibly difficult piece of the puzzle to isolate for many. So I'm glad that we can be inclusive for those who didn't have that confirmation. What is the ultimate goal of this study? What do we hope to take away from it? And do you have a timeline for results?
Yeah, really great question. So the goal of our study is the way that we envision it overall is that we do believe that you have to be exposed to a virus. It's likely enterovirus. And that's a trigger for something that you have within your own body, specifically in your genes that we don't know what genes it controls at this point because we haven't identified it. And it's that interaction between that viral exposure and your host genetics that may preclude someone to having paralysis following that viral exposure. And we suspect that timing in terms of when you're exposed in your life also likely matters, which is why we see so many children that are affected. So our goal is to identify that set of genes or that gene, whichever way it works, that explains that interaction that precludes you to having paralysis. So that's our big question. It's not just who's going to get AFM, but really what's the gene, what is the mechanism that's causing you to have AFM? Because we've seen this before. We've seen this disease before. It's polio. And we were so fortunate with polio to be able to have a vaccine that prevented so many children from actually having paralysis following infection because we could protect them with antibodies. But what we never understood with polio was what the mechanism was with why people actually got paralyzed. And so that piece is really important because once this enterovirus goes away, who's to say something else won't take its place? Right? So our goal is to understand that mechanism so that we can hopefully figure out early treatments for children that are exposed, and maybe for some families understand who's at risk in their own families for potential paralysis in the future. You also asked me about timeline. So let me tell you that we are not on a set timeline in the sense that we won't end the study until we really can get some answers. We are supported internally by our own institutes, but we are also applying for grant funding. So that is something that slows us down if we don't have enough money to actually accomplish what we need to do. But we are really dedicated to trying to get to this answer. So we hope to achieve, we have about 100 families. Our goal is to get to 300 families, which is about half the number of known cases in the United States. If we can get to that number, we feel like we might have enough statistical power to make some inferences about what genes might be playing a role. I think I answered all your questions, but I'm happy to answer one that I might have missed.
Yes, I think you did as well. Thank you so much for that. I know you brought up polio and that we've seen this similarly before, and we were able to isolate and develop a vaccine. Do you think that this study will lead us closer to developing a vaccine for the virus, or in the case of AFM, viruses that are suspect in it?
Yeah, so I will tell you that our study will not likely lead us closer to a vaccine. Our particular study is focused on the host genetic response, but I do know that there are many investigators out there working to try to determine which virus or set of viruses is really critical, and that work will really lead us towards a vaccine. And that will take a lot of effort for people to develop something that we think will work across the population the way that the polio vaccines worked. So, simultaneous to our work, there are researchers working towards those efforts and trying to understand the viral aspects and what kind of vaccine might work.
Thank you. So with looking at the genetic component of the cell, um, will this help us identify if there's a likelihood for recurrence of AFM or other issues from viral illnesses if that genetic susceptibility is found?
I think so. I think it may give us, until we understand the mechanism, it's really hard to say if this is specific to a set of enteroviruses. It likely is. If there is one, we think it likely is specific. And what we don't know is if there's the potential for recurrence, if this has any effect long term. And for many families, as we tell the families that have enrolled in our study, we can't do much for your child that's been affected already because the damage often has already been done. But we could try to help understand and help other families or perhaps other family members that you have that have not yet been exposed, but that may also carry the same predisposition.
That's great information. Thank you. So with that, if you do find that a particular individual or family has that genetic susceptibility to AFM, would participating families be notified of that?
Yeah, so I pride myself that I think we have a really wonderful relationship with most of the families that have participated in our study. We recognize how much they've had to go through in the immediacy of a diagnosis and then really trying to find answers to what's going on with their children. What we will provide to the families is a global understanding of what we've learned. What we can't provide is individual data back about what your child may or may not have with regards to genetic variants. We are doing this under a research protocol, not under clinical protocols, and there are differences in how you relay information back accordingly. But what you could learn from this is if we find potential genes that are involved, we will let families know what our findings are. And then families could choose to get tested specifically for their child in a clinical lab. And then they would know what their child has and what any other family members have in their family.
Got it. That makes sense. Being part of that research protocol and um, the information likely being very highly protected. So that kind of leads into the next question that we received from our community, from someone whose family is reluctant to participate in the ongoing AFM genetic study because they're worried that their genetic information may fall into the wrong hands and be used against them in some way. How can this individual allay the fears of their family members?
That is such a great question, and one that I think we're constantly dealing with right now as we think about genetic data overall. Let me just say that we take every precaution possible here at Johns Hopkins in our study. So once a sample is collected, we extract DNA from it and we get that sequence data. And at no point in that process, as we sequence that sample, is any identifying information about the individual who provided it given to anyone outside of our immediate research team. So we have that information of whose DNA this is, but no one else knows and can link that information back. And then similarly, because this is any part of this could be federally funded, we are under a congressional mandate to share data. And that's so that we can get to answers faster than if all of us just sort of isolated, looked at our own work and sort of held on to it. It's sort of like when you were in kindergarten and you want to hold on to your specific toys and the teacher really wants you to build a big project, right, with your friends. It's the same idea. The argument has been that, you have time to look at what you've collected, but if you could share with a greater scientific community, it would help everyone to get to answers faster. And so as a result of that, we will share some of our data with the National Institutes of Health, but the way that that data is shared is, again, completely protected. So no names are revealed, no information about where people live is revealed. What you'd have is that there's a case of AFM, and that these are family members that are a part of that. We do everything to sort of take away any measure of people being able to really identify you. So I hope that that allays some of the fears. I get, though, that some families are still going to be concerned. And if they are, I'm happy to talk to them and talk through our process and our procedures so that people understand what we do. All of our data is kept on secure computers and in locked cabinets. So for everything that we do in genetic work, but also just in general with people's clinical records, we do our best to protect them as if it's our own family or our own data.
Thank you. I know that that is something that is taken very seriously in the research world, and people are definitely protected by those rules and IRBs. So thank you for giving us more info on that. So with all of the information that needs to be protected, what exactly is it that is required of any one participating individual or family? How are you getting that genetic information? What medical records in particular? And how does someone start the process of collecting all of that?
Yeah, that's great. So we try to make it as easy as possible on everyone. We can do everything really by mail. That being said, if you happen to be in Baltimore, we are more than happy to come and meet you wherever you are. We like to meet the families, and we love to talk to you and answer your questions in person. The way that we do this is that a participant needs to reach out to us directly, and they can reach out to me via my email. And I'm happy to provide that after the fact if there's a way for you to share that. And once you've reached out to us, then we can contact you. We really can't reach out to you. So if you've been in the news or in the press, we can't reach out to you because of that. That's following our institution's rules. So once you've reached out to us, we will have a conversation with you, learn a little bit more about your child and your family and how AFM has affected you. We'll also gather some information on that call if you decide to participate about which hospitals you were seen at in the initial days and then post-diagnosis. And we'll find out information from you about who lives in the household, how many siblings there are, what their ages are, if mom and dad both live in the same household or they don't, if grandparents live in the same household. And then we send a kit out to you. It comes via the U.S. Postal Service. And in that kit are consent forms for every single person in the family that would be willing to participate. And that outlines all the things that we're going to do as well, very similar to the phone conversation I would have with you. In addition, there's a medical questionnaire. It's a really brief questionnaire that asks some questions about your family history, specifically getting at the child's family history and then the mom and the dad and any siblings. Initially in 2014, and even now, we don't really understand what other diseases might be associated with AFM or if there's an enrichment for a certain set of diseases. And so we ask some questions that might guide us. We ask about some autoimmune disorders. We ask about any other paralysis in the family. We ask if anyone's ever had polio in your family. And a lot of this is just information to help us just to try to see if there are patterns that are emerging among the AFM cases. And then also in there are medical record release forms. So from information that we gathered from our phone call, we actually have to reach out to those hospitals because most hospitals in the United States have their own forms. And we've learned that the hard way. So we will get those forms and we will have you fill them out and you have to sign it to allow us to access those medical records. Specifically, we're interested in the neurologic or initial diagnoses that happen so we can get a sense of how physicians viewed you as a patient in terms of your clinical symptoms, and then also imaging. So that allows those hospitals to release that imaging data to us. And then and then finally, I should say, the last thing is based on the number of individuals in your family that are participating, we send a saliva tube. So it's just a simple tube. You have to unscrew the tube if you're of an age where you can spit, and you spit into the tube. We recommend sometimes that people put a little sugar on their tongue or suck on a lollipop before. It just makes you salivate more. And then you just spit into the tube up until the fill line. And then when you close the tube, it allows a proprietary liquid to get released into the tube, which preserves the saliva so we can extract DNA. Each of those are placed in a special mailer, so in case they break open and spill, it'll be okay. And then all of that is shipped back to us in a prepaid mailer to our research study. So that is really the involvement. And at any point in time, you're welcome to reach out to us and to participate with us and to talk to us or ask us questions. But that's pretty much it.
Thank you. That's very helpful for anyone who may be considering participation in the study, what all it would entail and what they would be asked to do or to be prepared to collect. So a couple more questions from the community. Have you or will you be comparing any of the data and findings with research that may have been done on those with polio? And is there research on those previously diagnosed with polio that may help you in this study?
So again, a wonderful question from the community. So because of this AFM study that really emerged because of interest and because we thought we could try to help with our skills to answer these questions, we recognized that polio also didn't have any answers. A lot of work was done in the 1940s and 50s to try to evaluate polio cases. And one of the things that researchers noted at that time is that it likely did have a genetic component. They didn't have the technology or the ability to ask questions the way that we can ask questions now. And then, of course, the vaccine came along. And once the vaccine came along, many of us just I'm sure, ignored that question because it wasn't as relevant anymore. You weren't seeing, tens of thousands of children paralyzed every summer. And so that question got left by the wayside. And what we have from the literature is information that sort of leads us in the same direction. It is pretty amazing. I've seen some documents from over 100 years ago, led by Wade Hampton Frost, who was here at Hopkins, who evaluated one of the polio outbreaks. And the wording and the evaluation of those polio cases is almost identical to the way that we have looked at AFM. So it's very much as things have changed and improved on the polio front, we're really at the brink of eradication. We're seeing this new disease emerge that has such similarities, even in the way that it's been approached and emerged. So because of that, we have enrolled a separate study of polio participants. Many of them had contacted us because of media pieces that we had done about our study and about AFM in the big outbreak years, saying that they really wanted to talk to some of these AFM families because they had been through it themselves, that they really empathized with these children because they had experienced it. And so they had reached out saying, was there anything they could do? And so as a result of it, we started a separate study where many of these individuals in their 70s or 80s are sending us their DNA so that we can eventually compare their DNA to many of the AFM cases.
Wow, that's amazing. That's incredible of the polio community to reach out in that way too. So definitely so much to be learned from them. And one last question that we had is in the family members you have tested, have you found any similarities in those older ones that had polio and post-polio syndrome and the kids that have AFM today? Have you been able to look at anything there yet?
We haven't seen anything yet emerge. We don't have a lot of them sequenced, so we can't really evaluate in a systematic way at this point. That's our goal, but we're not there yet.
That makes sense. That makes sense. So I don't have any other questions. Is there anything that you would want to add that you would like a family to know before contacting you about the study or once they're involved in the study? Anything for them to keep in mind?
Not particularly. I think that, we have had such a wonderful response from families with AFM. I'm sure that there are many that we don't even know about that are either apprehensive about participating in research or really are just too busy with their child affected with AFM. We do our best to make this as easy as possible, and we really encourage you to talk to other families that have participated in this study, just to sort of see what you think might be helpful for you as well. Um, I'll also say that we are doing our best to come up with an answer, and we may not come up with an answer in the immediate future, but we just cannot appreciate enough how much the families who have participated have given to us, because I think they're really trying to help the greater good. They're trying to help the next generation, or the next 2 years of kids that might be affected. When I think back to many of the families we talked to in 2014, their statement was so clear, right? They knew that their child had AFM, but their effort was to try to prevent a child in 2016 from having AFM. And it's been every 2 years the same sort of story that we've heard. And that's actually what drives us as well. I will speak for myself and Dr. Millstone. Like, we are tired of seeing the headlines talking about a mystery paralysis. We really want to get to the root of this. And so, sort of larger participation from the AFM community, we hope will give us some answers.
Thank you so much. And actually, one other question that just came in too, for those who aren't participating, I know you mentioned earlier that you would let study participants know about what you found globally in the study. What about those who maybe are hearing this or are interested in the study? Where would they find and learn about the results of it, whether or not they are participants?
Yeah, that's great. So, because we're a part of a research community and this is what we do, everything that we do gets published and it gets placed in the public sphere of information. And so, whatever we find, we will publish and put in that sphere. And I imagine that once that happens, there will be avenues for us to disseminate information to the AFM community, perhaps through places like SRNA or others.
Yes, absolutely. When that becomes available, we'll definitely be sharing it through our resource library, likely our blog or newsletter um, as well. So, and if you would like, if you want to share your email address for those who may be interested in contacting you, and then we can also put a link to it on our website where individuals can email and find out a little bit more about the study as well.
Sure. So my email is P as in Peter, D as in David, U, G as in giraffe, G as in giraffe again, AL. So P Dougal at jhu.edu.
Wonderful. Thank you again for taking the time today. I sincerely appreciate it. I know many of our community members do. And if we do receive any questions, we'll make sure to reach out to you as well, and also continue to encourage folks who have been diagnosed with AFM. We know that it's not easy to reach out and to share your story essentially with someone you don't know, but as you've noted, it's so important to those who may come after us to help put this to an end. And if we can identify any factors, I think it's something that's certainly worth looking into. So thank you again so much, Priya.
Absolutely. Thank you so much for having me, and thank you for all that you do for the AFM community.
Thank you.